So now that we've kind of reviewed the different parts of the cells and we've talked about DNA and we've talked about chromosomes, what we're going to do is we're going to use that information to try and make predictions about the chance that a genetic event will occur. Now, for instance, we're going to look at specifically genetic disorders. So in your example that started us off this week, one of your parents has a genetic disorder that's called hemophilia. So you are probably asking yourselves, well, what are the possibilities or what's the probability that I might have a child that has the same disorder? And so this video is all about how we can use something called a Punnett square to kind of help us make predictions based on some basic principles of genetics and genes. So just to kind of go over what a gene is again, we've talked about already that a gene is a section of DNA on a chromosome that codes for a specific trait, right? So we have chromosome number 1. There's one tiny section on chromosome number 1 that say codes for eye color, right? And it's a series of letters, A, T, C, G, T, A, T, and that series of letters is going to be the gene, however long that particular gene is. When we're going to look at the factors that can make up a gene. So genes are kind of made up of two factors, which we call alleles. You get one allele from the egg, and you get one allele from a sperm. And then when they combine together, during fertilization, you end up with a gene that has these two different versions or these two different factors on it. Alleles can be dominant or recessive. So typically a dominant allele is represented by a capital letter, in this case an A, or a recessive allele would be denoted by a lowercase letter, a little a. Now, in most cases, whenever there is a dominant allele present, so this capital letter, that means that the version of that trait that the dominant allele codes for is the one that will be seen or the one that will be expressed. We'll talk about some exceptions to this rule, but typically we call this a dominant recessive relationship. So for instance, when we have two different versions of a gene, or two different alleles, we have three different combinations that could occur, okay? We can have big A, little a, which is called homozygous dominant. Homozygous means the same. Dominant meaning that they're both the dominant allele, so big A, big A. We could have a big A, little a. We got big A from mom, say, and little a from dad. This would be called a heterozygous dominant trait, or just heterozygous. Heterozygous meaning one of each. And then we could have a homozygous recessive trait, which is little a, little a. Homozygous, again, means the same, and then recessive, meaning that there are two little traits. In either of these situations, the dominant trait would be seen because there is at least one capital letter or one dominant allele. This one, even though it only has one, isn't any weaker or different than this one with the two capital letters. It's the same, but because there's a dominant letter, we would see the version of the gene that is represented by the dominant letter. This one does not have a dominant letter, so we would only see the version of that gene or the recessive version of that gene because there's nothing to dominate over. When we talk about genotype and phenotype, when we talk about genotype, we're talking about the combination of alleles. Is it homozygous dominant, big A, big A? Is it heterozygous, big A, little a? Or is it homozygous recessive, little a, little a? When we talk about phenotype, we're talking about what we see or what is actually expressed. So for instance, blue eyes or green eyes. In the examples we're going to do, brown hair or blonde hair, those types of things. So here is an example. We're going to start with a traditional dominant recessive example. So black hair here is dominant over blonde hair. So black hair would be represented by a capital B. Blonde hair is recessive, so it would be represented by a lowercase b, right? You have two of these letters. You've got one version from mom, one version from dad. You could be big B, right? So that would be homozygous dominant. That would be your genotype. and your phenotype would be that you had black hair. You could be heterozygous, big B, little b, in which case your genotype is heterozygous, your phenotype is black hair still, because there's still this dominant allele present. It's not less black than the other one, it's the same, it's black hair. And then you can be little b, which is homozygous recessive, and the phenotype for this would be blonde hair. The only way that you can have blonde hair, because it's a recessive trait, is if you have two recessive alleles. Okay, so you got a little b from mom and you got a little b from dad. There's no capital B or dominant version to kind of overtake this. So let's look at how we can use this information then to make predictions. So predictions will give us the probability that an event will happen. It's not a for sure thing. If there's a 25% chance that a genetic event occurs, that does not mean that 1 out of every 4 children all the time are going to end up with that genetic disorder. It just means that there's a 25% chance of it. You might have 8 kids with none of with the disease, or you might have 8 kids all with the disease, even if there's 25% chance. It's not a for sure thing. It's just an estimate, a probability, a prediction of what will happen. And we can use this a lot. Geneticists use this a lot, especially in genetic diseases, to give a couple who's thinking about having a children, like ours in our scenario, the probability that a genetic event will occur. Because depending on the severity of that genetic event and depending on what the probability is of passing that on to your offspring, it might sway your decision to choose to have children with, your sperm or egg and your partner's sperm or egg. So let's look at how we can use this thing called a Punnett square to figure out the genetic probability of something happening. So in this scenario, it's a classic dominant recessive trait. So brown hair is dominant over blonde hair. What is the probability of having a brown hair child if you are blonde, okay, and your partner is heterozygous for brown hair? So what we're going to do is we're going to figure out first what your parents are, right? So if you are blonde, so let's put the parent genotype here. Right? If you're blonde, you have to be little b, little b. It's recessive. It's the only way that you can have blonde hair. Your partner is heterozygous for brown hair. So remember, heterozygous means one of each, so it's going to be big B, little b. Now, we're going to put this information into this little square figure. So you're going to draw a box. You're going to divide it into 4. We're going to put one parent. We'll put this one. This was you along the side. Okay, and we're going to separate out their alleles, because when we talk next week about meiosis, this is what's going to happen during this process, where these alleles will be separated and it'll just randomly combine with the alleles of the other sperm or egg. We'll put this one up on top, so big B, little b. Now, all of these squares in here represent the offspring. So we're actually going to fill in the box then, based on what we have. So little b, big B, little b, little b, little b, little b. So you see that? This one came down here, this one came over here. This one is here, this one is here. Now, commonly asked question, does it matter if I would have written this big B, or big B? Does that make a difference? Nope, it makes no difference whatsoever. Okay, I typically put the capital letter first. It helps me kind of visualize looking for that dominant trait, but it makes no difference. All right. So these are the probability of our offspring. So let's go back. The question was, what is the probability of having a brown-haired child? So in order to be brown-haired, we need to have at least one big B. So is this one brown-haired? Yep. Is this one brown-haired? No, that one is blonde. It's only got 2 little b's. Is this one blonde-haired here? Nope, this one also only has 2 little b's. It's not blonde. This one has a big B, so it would be brown-haired. So there would be a 50% chance that these 2 parents would produce a child with brown hair, and a 50% chance that they would produce a child with blonde hair. So first, figure out the parent genotype, then fill out your box, your Punnett square, and look at the different offspring, and then 3, answer the question, whatever that might be. So here's another example. Try this one. Pause the video and see if you can figure out yourself, and then you can unpause it to see the answer. But again, we got brown hair is dominant over blonde hair. What is the probability of having a brown hair child if you and your partner are heterozygous for brown hair? Okay, so let's start with the parents. Parents are both heterozygous, right? So heterozygous is one of each. So we're going to have big B, little b, and you're both heterozygous, so it would be big B, little b. Okay, then you're going to do the square, and that was part 2, right? And here, okay, put this one along the side, big B, little b. We'll put this one along the top, big B, little b. Fill them in. Big B, big B, big B, little b, big B, little b, and then little b, little b. Answer the question. What is the probability of having a brown-haired child? So let's look. Does this one have at least one capital B? It does. So this one is brown-haired. Does this one have at least one capital B? It does. So this one is brown-haired. Does this one have at least one capital B? This one does. So it also has brown hair. Does this one have at least one capital B? No. So 3 out of 4, or 75% chance of having a blonde-haired child. Now again, just a probability. You could have 4 children and they could all be blonde, because blonde is still a possibility. It's just that there is a 75% chance that your child will be brown-haired. Hopefully that makes a little bit of sense for you. Now, there are some exceptions to this rule. Of course, it can't always be super easy, right? So the first exception to the rule is called incomplete dominance. And in incomplete dominance, what's going to be affected is the heterozygous genotype, right? So let's say big B, little b. Now remember, in a dominant recessive scenario, then the heterozygous genotype would show the dominant trait, okay? In this case, the dominant trait is weak and it can't completely overcome the recessive trait, so you end up with some kind of intermediate. So the best example that I can give you right now, kind of to make it a little easier to understand, is let's look at flowers, right? Flowers, a lot of flowers go through incomplete dominance. So let's say that red is the dominant allele for flower color, okay? And white is a recessive allele. So let's say we take a red flower, so red would be RR. White flowers would have the genotype little r, little r. Now remember, this genotype right here is what's different. So in the case of incomplete dominance, a big R and a little r would end up with an intermediate, which would give you pink. Okay, remember, in normal dominant recessive, this would be red, but because this is an exception to the rule, we end up with this intermediate color. Now, Huntington's disease is an example of this in humans, right? Huntington's disease is called by a dominant trait, so we could put HH would mean that you've had Huntington's disease. HH would mean that you were normal. But H, heterozygous, big H, little h, would give you a mild version of the disease. Okay, it's an intermediate in between this one and this one. Now, one thing I should have mentioned earlier, in the heterozygous situation, so this person right here, even though they might not have the disease, like in a regular dominant recessive situation, so let's go back actually and use this example. So these two, right, big B, little b, this person is considered a carrier. meaning that they carry the recessive disorder even though they don't show the trait of that disorder. Hopefully that makes sense. So here, this, even though it's exception to the dominant recessive rule, they would also be considered a carrier because they have that gene, even though they're going to have a much milder version because there's this intermediate here. The next exception is similar, except that instead of the dominant being a little bit too weak to overcome and you get an intermediate, in codominance, you end up getting you end up showing both traits. So in animals, this is what leads to being spotted, okay? In humans, this can be shown through a disease called sickle cell anemia, which is also recessive. So for instance, you have sickle cell anemia. It's a recessive trait, so this would be normal blood cells. Sickle cell anemia is a disease where the cells change shape. So SS would be normal. Little s would be sickle cell blood cells. And the heterozygous, right? So again, big S, little s, only affects the heterozygous. This would have both normal and sickle cell blood cells. Okay? So make sure that you know the difference between incomplete, where you get an intermediate, and then codominance in a situation where you would see both traits, both the dominant and the recessive trait being seen. And if you go back to this situation, in a lot of times in animals, they'll have codominant traits where you have like a red cow or red fur and white fur, and then this intermediate in codominance would give you red and white stripes. All right, the last exception to the rule is multiple alleles. These are traits that have more than two possibilities, but still only two alleles will be present on the gene. So human blood type is an example of this. There are 3 different alleles. Instead of a dominant and recessive situation, we have a different mixture. We got an A allele, a B allele, and an I allele. Sometimes it's written as an O allele as well, so you might see it written a little different. But if you have blood type O, you can be either big A, or you can be big AI. Blood type B, or big BI. Blood type AB, you can only have an A and a B. And blood type O, you can only have 2 O's. So in this situation, it says, can a blood type BI father, so let's make our box here, okay, BI father, and an AA mom, so she's blood type A, produce a type A child? Well, let's see. This offspring would be blood type AB. This offspring would be AI, right? I and A. This one would be AB, and this offspring would be AI. So this is blood type AB. This is blood type AB. We want a blood type A child. This is an A child, and this is an A child. So there would be a 50% chance of having a type A child. So same general ideas, you just got to be careful. So if I would have worded this question and said a blood type B father, you'd have to do 2 different Punnett squares because you wouldn't necessarily know if it was BI or if it was BB. All right, last example then are going to be those traits that are sex-linked. So some genes are found on the X or Y chromosome, and this is actually going to be your hemophilia example. So because they are attached to the X or the Y, one of the sex chromosomes, they're going to affect males and females a little differently. So when you do sex-linked traits, you have to include the X and the Ys on there in order to see whether or not females or males will end up with that disorder. So for example, hemophilia is a sex-linked recessive trait. What is the probability of a hemophiliac male? Okay, so let's start there. So we know that a male is going to be XY, right? And this is, I'm sorry, I should have written this on here, but this is going to be an X-linked trait, hemophilia. So if it's a hemophiliac male, and we know it's recessive, then this has to be a little h, right? X-linked means it's found on the X chromosome. It's not found on a Y chromosome, only on the X chromosome. So males only have one X chromosome, so if they have the disorder, then this has to be recessive, right? Because if it was dominant, then they would be normal. There's nothing to, overcome that. But you got a heterozygous normal female. So let's put a female here. Females are XX, right? That's their 23rd chromosome. A heterozygous normal female means that heterozygous, there's one of each. So she's got one dominant allele and one recessive allele. Now, she would not have hemophilia. This person does not have hemophilia because there's this dominant trait that's, taking over. It's just like a regular dominant recessive relationship. But when we fill in these boxes, you're going to see what happens here. So here's X big H. Here's X little h. We'll do the girls first, the daughters. This is going to be X little h. This will be X little h. This daughter will have hemophilia. Look at the Y's, the boys that will be produced, right? You got XY, X big H. So this son will not have hemophilia. This son, X little h, will have hemophilia. So there's a 50% chance that their children in general will have hemophilia, but it says what will be the hemophiliac daughter. That's a 25% chance. There's a 25% chance that a girl here will end up with hemophilia. This person is a great example of a carrier, right? They're normal, they don't have hemophilia, but they carry this recessive gene that can cause hemophilia. And if it mixes just right in a given situation, it can lead to the development of the disorder. So you're going to be looking at this in your assignment today on how this actually works when you have a parent, like one of you or your partner has a parent who has hemophilia. What is the probability that your children then will have this disorder? There are videos posted in the resource folder to help clarify these even more if you need it, so please don't be afraid to ask, and don't be afraid to ask me questions. This can get a little bit complicated, and we're going through it relatively quickly for the sake of time. So have a great day.